from the biochemistry question paper, GATE Excel 2021, which means Life Sciences Biochemistry question paper. Question number 8. So which says that which of the following enzymes is used in polymerase chain reaction? Clinofragment, Taq polymerase, T7 polymerase, primase. So this is a for following more details in Taq polymerase, you can follow this particular genetic engineering theory and applications NPTEL course for more details on polymerase chain reaction. Now we will first look at what are some of the components in the polymerase chain reaction. So the first one is, for example, the first one is the sample DNA, which is our template DNA. Sample, or which is called the template DNA. Now the next one are the primers. So these DNA primers are what? Primers are oligonucleotides. So these are oligonucleotides. These are usually short oligonucleotides which are complementary to the template DNA. So there are two types of primers which are used, the forward and the reverse primers. So we will look at them in more details in the later slide. And now we also have this important component, which is the nucleotides. So these nucleotides will help in polymerization of the new daughter strand, right? So therefore these are the raw materials which will polymerize to produce the amplified DNA sample. Okay, and the next one is the enzyme, which is the Taq polymerase. You have guessed it correctly. So the Taq polymerase is an enzyme which is a thermostable DNA polymerase. So we all know about DNA polymerase. What is a DNA polymerase? It polymerizes the synthesis of a new DNA strand based on the template DNA strand. And this enzyme, this particular enzyme Taq, it is a special enzyme. Why? Because it is a thermostable DNA polymerase, which means that it is stable at higher temperature. We will discuss Taq polymerase again in detail. And we also have a buffer. So this buffer will have the buffer components as well as the required salts. And we set up this experiment in a tube which is called a PCR tube. So this is a very small tube of 0.5 microliters volume. So in this tube we set up this entire experiment, and then we run this experiment in this machine which is called a thermal cycler. So what is a thermal cycler? It is a machine which actually regulates temperature. So in this machine you can set a reaction, which means that you can set for example, at what temperature the denaturation process of DNA will occur, then at what temperature the annealing of the DNA will occur. So this temperature you can regulate in this particular cycler, thermal cycler, and this is called cycler because it can cyclically regulate the temperature. Now this is these are the components of the PCR, that is the polymerase chain reaction. Now what are the steps of polymerase chain reaction? The first step is denaturation. So what is this? So this is heating the DNA at very high temperature so that the two strands of the DNA can separate. So this is at 95 degrees centigrade. Okay, so which means the DNA strands separate and the double stranded DNA becomes single stranded DNA. Why do we do that? So that our primers can go and sit on the complementary template DNA. right? So these are our primers. This is the reverse primer, this is the forward primer, okay? And these primers, in order for them to sit on the DNA has to be denatured first. It has to be made single stranded first. So that is what was done. The DNA is denatured. Then the temperature is maintained in which the forward and the reverse primers can anneal to this template DNA, right? So this is called the annealing step. And next, so we can see it can range between 55 degree to 50 degrees. So the annealing temperature depends on the sequence of the template DNA as well as the primer DNA. And the last step is the extension step, where the primers now start extending. So it starts the synthesis from the free 3' hydroxyl end. So this is called extension, and this extension temperature varies on the DNA polymerase that we are using. Usually for TAC, it is in the range of 68 to 72 degrees centigrade. So these are the components of the PCR reaction and also the steps of the PCR reaction. Now let's look a bit at the TAC polymerase. So the TAC polymerase is the most common enzyme which is used for PCR amplification, and it was first discovered in this thermophilic bacterium Thermus aquaticus. So this is a bacteria which is found in the hot springs, and this bacteria, it can survive in this hot spring area. So it was a question how this bacteria can survive in these very hot conditions, at a very high temperature, because we know that the enzymes or our proteins, they are sensitive to high temperatures, because at high temperatures the proteins lose their three-dimensional structure, right? So therefore, if they lose their three-dimensional structure, they will also lose their function. So proteins are sensitive to heat. Now therefore, at this high temperature where this thermophilic bacterium can survive, how are its proteins designed, how are its enzymes designed so that the proteins and the enzyme can tolerate such high temperature? So it was discovered that this kind of bacteria, which is called Thermus aquaticus, it contains a DNA polymerase which is called after the name Thermus aquaticus. The T is taken from Thermus and AQ is taken from aquaticus, and thus the name of the polymerase was given, TAC polymerase. And this is a common DNA polymerase of this kind of bacterium. Now this enzyme is extremely heat resistant with a half-life of 40 minutes at 95 degrees centigrade. So it has a half-life of 40 minutes at 95 degrees centigrade. So at 95 degrees centigrade it can sustain for a long time. And what is its optimum temperature? The optimum temperature of this enzyme is 72 degree, where this enzyme can synthesize this polymerization, or it can perform this polymerization, it can catalyze this polymerization reaction, and here the nucleotides are incorporated at a rate of 2 to 4 kilobases per minute. So this is the rate of nucleotide incorporation of the TAC polymerase. Now this enzyme also catalyzes 5' to 3' synthesis of DNA, and it has no detectable 3' to 5' exonuclease or proofreading activity, and it possesses low amount of 5' to 3' exonuclease activity. So this is the naturally available TAC polymerase, but however TAC polymerase now has been designed in the labs to produce for higher fidelity, for higher processivity, right? So TAC polymerase also exhibits deoxynucleotidyl transferase activity. Deoxynucleotidyl transferase activity, what is this? It is activity which results in addition of extra adenine at the 3' end of the PCR products. And recombinant DNA polymerase, TAC DNA polymerase, what I was talking about, that nowadays the TAC polymerase is engineered for PCR reactions, such that it has low error rate, higher processivity. So this recombinant Taq polymerase is a very good enzyme which we can use for standard PCR of templates which are 5 kV or shorter. Okay, so for shorter DNA it can be used with high efficiency, but for longer DNA, for PCR of longer DNA substrates, we usually need polymerase which has higher proofreading activity. So low error rate is required for amplification of longer DNA molecules. Now these are the primers that I also wanted to discuss about in this question, that the forward and the reverse primers. So the blue strands are the DNA template strands here, and the red one are the primers. So I told you that there are two types of primers, the reverse primer and the forward primer. Now it is very important to understand their orientations. So if we look at these template DNA strands, one is from 5' to 3', another is from 3' to 5'. Okay, and why is that so? Because we know that two strands of DNA are anti-parallel to each other. So one is 5' to 3', another is 3' to 5'. Now the forward primer, where does it sit? What is the utility of the PCR forward primer? The PCR forward primer sits at this particular site. This is the site where the PCR forward primer sits. And what is its role? So you can see that it is complementary to this region of the DNA template, right? And it synthesizes. So this end has a 3' hydroxyl free end, from which the polymerase can start synthesizing the nucleotides, because it always provides a free 3' hydroxyl end. Here also, in case of reverse primer, now note that the reverse primer sits in an opposite orientation, because the reverse primer sits on the other strand of the DNA, right? It is complementary to the other strand of the DNA, and you can see that it is complementary to this particular region of the template DNA, and it sits in this particular orientation from 5' to 3', right? So this is the this also provides a 3' hydroxyl group, from which the polymerase can keep on synthesizing or adding new nucleotides, right? So we have these components where we have the DNA template. We add the DNA template with the primers. Now the primers, first of all, we denature the DNA templates so that they become single stranded. Then we add the primers. So what is the when we add the primer, it is called the annealing. And then what we do is we extend this, we allow the enzyme to extend this newly developed DNA strand, right? So that is called extension step. So here also remember that in PCR amplification or polymerase chain reaction, it also occurs by exponential amplification. So suppose this is the template DNA that we have started with. And this red portion is our wanted gene. Now how many molecules of DNA will be produced? This also comes in some questions, so be very careful about it. How many molecules of DNA will be produced at the end of each cycle? So after the first cycle, so if we start with 2 molecules of DNA, after the first cycle we get 2 to the power 2. That means 4 copies of the DNA molecule, right? So next is after the second cycle we get 2 to the power 3, which means 8 copies of the DNA. So we can see there is an exponential rise. After that we get 2 to the power 4 after the third cycle. After the fourth cycle we get 2 to the power 5, which means 32 copies or 32 molecules of DNA, right? So this is how the exponential amplification goes on with the help of this Taq polymerase. So that is the role of this polymerase. This exponentially amplifies the DNA, and also it is extremely heat resistant. So when we heat the DNA samples at 95 degrees centigrade, at this temperature, this particular enzyme does not have any problem, right? So it does not face any degradation. So therefore this is an ideal condition where Taq polymerase can be used. So that is it. So coming back to the question once again, therefore the right answer is Taq polymerase. Which one of the following enzyme is used in polymerase chain reaction, which is called the Taq polymerase? So